Good afternoon, everyone. Thank you for joining us today. My name is Casey Zia, and I'm the Associate Director of Donor Relations and Communications. Before I introduce Allison Purvis, the Office of Development's Chief Development Officer, I wanted to go over the flow of the meeting and how we're going to handle questions. Everyone is currently muted, and that is just to make sure we can hear everyone's questions and individuals are not speaking at the same time. We want to make this an interactive meeting, so as you have questions, feedback, or comments, please use the raise your hand feature, which can be found under the reactions button. Once you raise your hand, I will unmute you and you can ask your question. You can also type your question in the chat box and we will address those one by one. If you're having any technical difficulties, for example, you can't find the raise your hand button, please send a private chat to Moses Ramirez. This can be done by clicking the chat box at the bottom of your screen. Once that opens, at the bottom of the window, you will see a box that says to and a down arrow. Click the down arrow and Moses's name will be an option. Don't forget to change it back to everyone if you have a question for the speakers. At this time, I will hand it over to Allison.
Great. Thank you very much, Casey, for pulling this meeting together, one of our donor Tele-Townhalls, special events put on by the Office of Development in recognition and thanks to you, our audience, and our important partners in philanthropy at the academies. We're thrilled to welcome you this afternoon on a special occasion to hear about the COVID virus variants. This is a panel hosted by our President of the National Academy of Medicine, Dr. Victor Dzau, and I will now hand it over to him to introduce our panelists and begin what we hope is a warm, engaging, and informative session where you're invited to ask questions and participate after we hear from our esteemed experts. So thanks very much, and over to you, Victor.
Thank you, Allison. Welcome, everyone, and it's great to see all of you here. And I see that my friend and colleague, Marsha McNutt, the president of NAS, has joined us as well. Welcome, Marsha. When we thought about doing this Tele-Townhall, the staff had a lot of great ideas, but it was prophetic when I said, let's do the variant. And at the time, we didn't have Delta, but already I thought variants and vaccine is a big topic. And so I'm so glad we picked it. And the fact that so many of you are joining us is to show how important this topic is. We're so lucky to have two world-class experts on this topic. You couldn't ask for more. I've worked with them, I've attended meetings with them, and they're true experts. Let me first introduce them as we get into the whole conversation. Angela Rasmussen. She is a virologist at the Vaccine Infectious Disease Organization, the University of Saskatchewan. She's an affiliate of the Georgetown Center for Global Health Science and Security. Her research focuses on host response to infection with the emergent viruses, including SARS-CoV-2, and how viruses interact with the host to cause disease. She had just moved to Canada. I know she's highly sought after by the Canadian news media on this topic, and she indeed is a great expert. So welcome, Angie.
Thank you so much for having me, Victor.
Great. And the other person is Carlo Del Rio, a good friend, International Secretary of the National Academy of Medicine, and Distinguished Professor of Medicine Division of Infectious Disease at Emory University School of Medicine. He's also Professor of Global Health and Epidemiology at the Rowling School of Public Health. He's also Executive Associate Dean for Emory at Grady and the Principal Investigative Co-Director of the Emory Center for AIDS Research. I can read on, but if you ever watch television, you know he's on television in CNN and others frequently. So certainly during the COVID, he's played a major leadership role in designing response, speaking publicly, publishing important papers. And so we're so glad to have you as well, Carlos. Welcome.
Happy to be with you, Victor, and with members of the, donors to the Academy.
Great. So I should say this group is very special to us, not only speakers, but the donors. You help us do what we can do. In many of our initiatives, it's with your help that we're able to carry them on and make a difference. So thank you. So let me just frame our issue very briefly, because I really want to spend more time having a discussion among the three of us and then open for general Q&A. You all know where we are today. Well, a year and a half into COVID, we now have about 3.8 million deaths in the world and over 600,000 deaths in the United States. And I'm afraid to say, Angie, I don't know what the number in Canada is, but it's high anyway.
It's high relative to the population of Canada.
So, I think that while we are seeing in US COVID cases falling and rates of deaths and hospitalizations decreasing, and we now know we have effective vaccine, I think we're not in the clear. Well, the good news is at least the vaccination effort US is going quite well. 64% adults receive at least one shot. 86% of people older than 65 has been vaccinated. 50% and more are fully vaccinated. And our President Biden has set a goal for 70% of population to receive at least one dose by July 4th. So this is moving in the right direction. But we do know there's still a lot of vaccine hesitancy, and there's still a long way to go, particularly in some of the states. But my feeling is as we're thinking better and life is beginning to return to normal, we're not out in the clear. Even though our cases are dropping, we can see they're starting to plateau. And vaccine campaign has slowed down. And we're still dealing with a substantial number of Americans hesitant to receive vaccine. Then we now have the variants. The concerned variants have really upped the game, if you will, because the uneven rolling out of vaccine in certain areas in the United States and certainly globally. In Africa, only 0.6% of Africans are vaccinated. India, 3.5%. Brazil, 11%. We've got a long way to go. And the question about vaccine, the variants is, what are they? Are they going to, in fact, outrace the ability to vaccinate? That's the conversation we're going to have today. Just to tee it up, right now we know quite a few variants, but the ones that's being talked about are alpha, beta, gamma, and delta. Alpha is the one that started in the United Kingdom, and I'm sure my learned colleagues will tell you about what the characteristics are, but they're more transmittable. South African variant of beta is probably even worse, and Brazil, and now we got the delta from India. I just want to tell you one thing, and I'm going to turn to them. When UK started this, and they started feeling really good about themselves, they've seen a recent surge, and it's probably attributed to Delta because right now, 91% of new cases in UK are Delta. And United States, 10%, but in some of the Western states, it's up to 20%. So let me then turn to my colleagues to say, tell us about what is a variant. I mean, people want to know what a variant is, and then how do you track and identify that the variants? How do you detect the emergence of new variants, and what kind of surveillance system? Let's start with that first. Angie?
Yeah, I feel like this is a question for me. So variants, variant is actually a very generic term that really just describes a virus that acquires mutations from the parent virus. So we've actually seen variants emerge throughout the entire pandemic relative to the prototypic Wuhan 1 strain that first emerged in early 2020 in China. So basically, as the virus replicates, it will make mistakes. Some of those mistakes will be preserved in its genome. And for the most part, a lot of those mistakes or mutations don't really have any effect. Many of them are what are called non-synonymous, meaning that they don't even result in a coding change or a change in the protein where that mutation is occurring. But every once in a while, the virus will sort of win the evolutionary lottery and have one of those mistakes be made in a place that gives the virus some kind of advantage. And the more the virus has an opportunity to replicate, the more of these mistakes it will acquire. Essentially, each time it replicates, it's like buying a lottery ticket. If it buys enough tickets, it's going to win. And that's exactly what's happened. So the first variant of concern, even though we didn't call it that at the time, was actually one called D614G. That was a mutation in the spike protein. And now essentially all the viruses worldwide that are circulating have this D614G. It was thought at the time that it makes the virus more transmissible. It's still not clear if that's what's happening in the real world, but it definitely provides the virus with what's called a fitness advantage. That means that the virus can replicate more robustly, potentially leading to more shedding, and it makes it more infectious or infective, actually. That means that it binds its receptor more tightly and is more capable of entering cells. So the variants of concern that we've observed now are among this huge pool of variants. And they are all variants that have these qualities. Either they have enhanced transmissibility, or they have the ability to partially evade immune responses against the virus. Unfortunately, we still actually don't know much about the mechanisms of this. We do know that Alpha, the variant first discovered in the UK, and Delta, the one first discovered in India, both are more transmissible. Beta and Gamma, first discovered in South Africa and Brazil, appear to be more capable of evading immune responses. But fortunately, the more transmissible ones, Alpha and Delta, are still, the vaccines, at least full vaccination, does provide really robust protection against those. The way that we're going to be able to detect new variants emerging is through something called genomic surveillance. That's when you collect samples from people who suspect that they have COVID-19. Instead of just doing PCR, which is the standard molecular test for diagnosing somebody with COVID-19, you actually take those samples and you fully sequence them to see if there are intact viral genomes there. By analyzing those genomes, you can determine if there's a new variant in town. But even that won't necessarily tell you if you have a new variant of concern. The reason why B117 or Alpha was detected in the UK is they have excellent genomic surveillance there. And what they saw was not just that this variant was emerging, but that it was emerging rapidly and overtaking all of the other variants in that community. That's the same thing that we've seen with Delta, and that's what we're going to need to watch out for in the future in terms of identifying other variants of concern that will emerge.
So Carlos, in this regard, we know we did not do a lot of genomic surveillance, at least not recently, right? There's the argument whether we have the system, we have the capacity, not like UK. What do you think of where we are today, and how are we overcoming this problem?
So I think we are, Victor, but I think we were late. I think we were way behind in genomic surveillance. And again, part of it was because of many, many years of underinvestment in public health. We didn't have the equipment necessary. We didn't have the personnel necessary to do this. So at the, even as recent as, November, December, we were sequencing probably less than 10% of isolates that were found. We were like number 48 in the world in percent of isolates doing genomic sequencing. And obviously, if you don't know what is circulation, you don't know, right? You don't know what you don't know. So I think one of the most important things that has been done recently since the arrival of Rochelle Walensky to CDC is really try to build robust genomic surveillance at the state level and at CDC. And I think right now we are beginning to have a very clear picture of what's going on that allows you not only to see what's going on, but actually allows you to predict what's going to happen. I mean, it was very clear that looking at the data, Dr. Walensky was able to say by March 23rd, the Alpha variant or the UK variant B117 will be the predominant variant in the US, and that was indeed the case. So they're able not only to have the surveillance, but also to make the modeling to predict what's going to happen based on the reproductive number, based on how frequently, the percentage is multiplying over time, et cetera. So I think building capacity for surveillance is something that is very important. But again, when you think about this pandemic being global, we really don't know what's circulating in many places. And sometimes you find out when somebody from India travels to the UK and surveillance happens there. Somebody from the Brazil variant was brought into attention when there was an outbreak in Vancouver related to the P1 variant. So I think that building global genomic surveillance is going to be important as we tackle this pandemic from a global perspective.
Yeah. So Carlos, is it not true that, first of all, the testing is really a local state issue to begin with, maybe coordinated by CDC, and the surveillance is not as systematic for the entire country, right? And is that not the case at this point?
Yeah, I think it continues to be the case. I think what's happening that I think something we need to be aware of is that increasingly a lot of the testing, rightly so, is being done through antigen detection, not necessarily PCR. And as you go more into antigen testing, you have less capability to do the genomic surveillance. So you need to almost think about having the antigen testing as point of care or diagnostic purposes, and then being able to come up with a way that you set up the surveillance systems in place so you can actually get some PCR, get some isolates, and be able to do surveillance, because otherwise you're going to be left without the ability to really know what's going on. Yeah. So I think it's really building both diagnostic and surveillance capacity almost in parallel.
One thing that came out from this experience, I know I've worked with the academic medical centers like yours, Vanderbilt and others, is that there's capacity in the academic centers, but CDC and others are not connecting with them, using them. So in fact, the group of leaders in the academic center with me and David Scott talk to CDC and say, look, we can give you that capacity. In fact, we need to link together the data and the information. We can become regional hubs to solve this problem for you. And their response was, we don't have the money to support what you do. So to your point, I kind of, but we are missing that opportunity.
I think we are. And I think it's an issue of really, public-private academic partnership, because remember, a lot of the testing now is being done by Quest, by LabCorp, by all these organizations, and really bringing all that data together and doing, big data analysis and being able to look at all that together, I think really is what's going to make a big difference. It's beginning to, but I think we have a long way to go to actually get there.
Angie, coming back to the variants, can you walk us through the 4 variants that we talked about, or maybe the 3 ones that's particularly of concern in the United States? A little bit more detail about what's alpha, what's it like, what about beta, what about delta? And then I'd like to talk about vaccines, because people are really worried about, okay, if I've got it, what does that mean if I'm vaccinated? So walk us through the characteristics of those 3 or 4 variants.
Yeah, so all of these variants have mutations in the spike protein, which everybody, I think at this point, has heard about. That is the protein that's on the surface of the virus particle that binds the receptor ACE2 and allows the virus to actually get into a cell and infect it. The spike protein is also what all of the vaccines are targeting. It's the major antigen that neutralizing antibodies at least target to render the virus non-infectious. So those are the mutations that we focus on, but actually all of the variants do have mutations throughout the genome, and we don't know very much about what those other mutations might do. They certainly might have an impact in terms of viral fitness, as well as potentially circumventing other parts of the immune system that are not antibodies. So T-cell responses and innate antiviral responses, things like that. But we don't know much about that. So most of what has been done has been looked at to characterize the mutations in the spike protein and to understand what they do. And we know from epidemiological data at this point that both alpha and delta are more transmissible. We don't entirely understand the basis for that. It may not, as I mentioned, be just because of the spike protein. And this transmissibility can happen, increases in transmissibility can happen in a few different ways. It can be the result of an increase in viral fitness. That means that basically there's more virus being made. The virus is infecting cells, and more infectious virus is being shed from those people who are infected. It could be the result of increased infectivity. That means that basically the spike protein is more capable of binding ACE2, the receptor, and therefore it's more efficient at infecting and entering cells. It could also be, although we don't really have any data to suggest that it is, that the virus is more stable in the environment for longer periods of time. So you'd be more likely to become infected if you were exposed to it, either in the air or potentially in close contact with another person. It's also possible, there was one very, very small study that I don't think we can make any conclusions about with alpha, that looked at NBA players, that suggested that they had a longer period in which they were contagious. So it could be any of these mechanisms that are leading to increased transmissibility, or a combination of them. We know from in vitro data that at least the alpha variant is more fit in cell culture. It does grow to higher titers. We also know that for alpha, beta, and gamma, actually, they are more infective. There's a mutation that's common to all three of them, N501Y, which is in the receptor binding domain, the part of the spike protein that meets up with ACE2 that increases affinity, meaning that the tightness that ACE2 binds to that. And we know that sometimes combinations of that N501Y mutation with other mutations in the spike protein can actually enhance that further. So we know a little bit about what these mutations are doing, but we still don't really understand the basis for which they are increasing transmissibility, because B1351 or beta and P1 or gamma don't seem to be more transmissible, but they are more capable of evading at least neutralizing antibody responses. Again, that is determined in the lab. Yeah. It does look like in fully vaccinated people, those vaccines are a little bit less protective against these variants, but it's really important to note that we have not yet seen a variant that can completely evade any of the vaccines. The vaccines all provide robust protection against all the variants in terms of preventing severe disease. The thing that's most worrisome to me about Delta is that not only does it appear to be more transmissible than Alpha, and again, this is based right now primarily on epidemiologic data and molecular epidemiology data, which is a combination of epi and the genomic surveillance that we were just talking about. It seems to be even more transmissible. So it's the most transmissible one we've seen yet. And also, while full vaccination does provide almost the same protection as against any other variant, partial vaccination does not. And that's particularly a problem here in Canada. Now, I was fortunate to be fully vaccinated before I moved here. But Canada and the UK both decided to take a different path than the US, and have recommended a delayed interval before the second dose of the vaccine. And now, fortunately, at least in Canada, our supply is increasing. So they're really encouraging people to get their second shot as soon as possible, because even with small amounts of Delta, we've seen how in the UK, before that in India, this variant took over very quickly. So it's going to be really important to make sure that people are fully vaccinated to keep this variant from spreading further. Because the other thing that we've seen among these variants is something called convergent evolution. We've seen some of these mutations in multiple variants, and they clearly didn't emerge from each other. What's really worrisome, though, is that if these variants are allowed to continue replicating, if they continue spreading into unvaccinated populations, they will potentially acquire more mutations, including combinations of some of these mutations that we think are contributing to their spread and potentially to more vaccine evasion. So it is really important to get people fully vaccinated while we know the vaccines are still protecting against these more transmissible variants.
Thank you. Well, you kind of said at the very beginning, I mean, they are undergoing mutation and there's a selection pressure. I think to a large extent, the immunization is a selection pressure for those variants. that escape immunization or in fact, in the current environment. So we are worried that one of these variants is going to totally escape immunity, at least conferred by the current vaccines. Carlos, can you expand on this? This is a very important discussion. Currently, our vaccines are very effective against the original SARS-CoV-2, right? Some 90-some% protection. And then, if you what is the data? Angie kind of talked about this of Moderna, Pfizer, AstraZeneca, all the others against some of these variants. And let's talk about Alpha and Beta and Gamma first, because Delta, I think, needs to be treated somewhat separately.
Well, I think what happens is clearly not all vaccines were created equal. And what I mean by that is they were not tested at the exact same time. So when you look, they're not tested in parallel. So you really have a lot of trouble making a lot of saying a lot of things. We know that Pfizer and Moderna, when they were tested, the predominant strain was the original strain, the parent strain, and that's where the 95% data comes. Had Pfizer and Moderna been tested today, maybe the results would have been different. When you had, for example, AstraZeneca, when it was tested, and I'm thinking about, for example, when it was tested in South Africa, there was very clear evidence that it didn't have much impact against the South African variant, but it had good impact against the others. So, but you cannot say one thing or the other because they were never been tested together. What we're relying a lot on right now is sort of not, efficacy studies, but effectiveness studies. We're looking at things that happen, for example, in post-approval studies that are being done, and a lot of it also happening in the lab. In laboratories, people are looking at, can this antibodies produced by this vaccines neutralize this or that strain? And in that respect, it's pretty clear that the mRNA vaccines are very good at neutralizing Alpha. And Alpha, again, right now is the predominant strain in the US. As I said, March became the predominant strain in the US. And now 75% of isolates roughly in our country are the Alpha strain. And both vaccines are very effective against that strain. The Johnson & Johnson vaccine is equally effective against that strain. So we should not have a problem there. Now, when you're talking about other vaccines, you're talking about AstraZeneca, fairly good efficacy. But when thinking about some of the, for example, CanSino or Sinovac, which are inactivated virus vaccines, we're not very sure. And for example, we know countries like Chile that have vaccinated a lot of people with some of the Chinese vaccines. They're beginning to see they never were able to control this strain. So is that because they had problems with vaccination, or is that because of problems with the efficacy of the vaccine? So for example, Bahrain has decided that since they have a large percent of the population vaccinated with the Sinovac vaccine, they have made the decision that because of the emergence of some of the variants, they're giving everybody another shot now of an mRNA vaccine. So we're beginning to see mixing of vaccines as a way to sort of try to address some of this variant issue. So when people say, well, I'm vaccinated and I'm going to travel to X country, I think it's very important to know what you were vaccinated with and which country you're going to. But the opposite happens as we bring students from other places in the US, or we bring workers from other places into the US. Just because they're vaccinated, it may not be sufficient. They may need to be vaccinated because the vaccines may not be the same. As I said, it's very different, the FDA approved vaccines versus what the WHO has in their list. Now, when you're talking about the Delta variant, I think what Angie mentioned, and I want to emphasize, is that you really need, even with Pfizer and Moderna, you really need 2 doses of vaccine. And you mentioned at the beginning that President Biden has made the commitment to have 70% of Americans having received at least 1 dose by July 1st. I really wished it was really, we bumped it up to say, stop talking about 1 dose and say fully vaccinated, because by July 1st, the Delta variant is going to be predominant in this country. And if we don't go to full vaccination, we're not going to have the level of protection that we're hoping. And in fact, many states are beginning to see upticks in their case rate. And it's because their vaccination rates are not very high of fully vaccinated individuals. So really, we need to really emphasize this concept of full vaccination in order to be fully protected.
Well, truth is, early in this, when the vaccine's available, there's not enough. And that's why Canada and UK decided to do what we call dose delaying or dose stretching approach, right? And it's still, I think, reasonable to say, if you get most of people having 1 dose, you have pretty good coverage. with exception that now you have Delta. So I don't think it's a fully incorrect strategy, but it may be insufficient, I would say,
Correct.
right? And as you know, there are people out there, including one major Nobel laureate in economics I work with in the G20, where they think about dose fractionation. To cover the rest of the world, why don't we split the doses? Because we never really know what dose is enough, what's not. And that you should do some pilot to look at how you can stretch your dose. So Angie, let me ask you this question. What is the rationale for, if you take Delta, that you need 2 doses? Does it mean that first dose, you're not getting sufficient immune response and you need the second dose to boost it up? And what about all those vaccines that you only use 1 dose?
Yeah, so let me start with why is the second dose needed? That is thought to be needed for affinity maturation, which is the process by which, so actually, let me back up. When you get that first dose, your antibodies, the B cells that make those antibodies and your T cells will recognize that the spike protein that you are getting, either from AstraZeneca or from mRNA vaccines, is foreign, and they will start making antibodies. Now, your immune system normally has this process where it begins to make these unique antibodies, but then it can actually edit those antibodies to make them even better, especially when it comes to antibody neutralization. And that process is called affinity maturation. It's basically like taking an introductory class and then you take the advanced class. And that's essentially what's happening with your immune system. So it's thought that after the first dose, even though you do have some protection, you don't necessarily have that lasting effective protection until you get the second dose, and you can basically instruct your B cells to start undergoing this affinity maturation process. It's also likely that this also is telling your CD4 positive T cells that they need to start instructing your immune system to develop long-term memory. So that is the reason for the second dose. It also explains why the first dose is partially protective, but not fully protective, especially over a long period of time. And as you mentioned, the dose stretching strategy was really based on some very preliminary data from the clinical trials that showed that there was protection 2 weeks after the first dose. But because those trials were conducted as part of 2-dose regimens, we don't know how long that protection lasts. So that was the risk with that strategy. And it worked pretty well up until the Delta variant came out.
Yeah.
Now, in terms of 1-dose vaccines, Johnson & Johnson is a different type of vaccine. It's an adenovirus vectored vaccine, which means that there is a virus that is undergoing at least part of its replication cycle when you get that vaccine. That is thought to potentially lead to more robust T cell responses, which could trigger that affinity maturation process in 1 dose. But the reality is we don't actually know. We think that Johnson & Johnson is still very protective against all the variants in terms of severe disease, but we actually don't have a lot of effectiveness data in terms of how well it can prevent infection overall, and it may not. And I think that one thing we might be seeing going forward is a recommendation that people who have had Johnson & Johnson, like myself, end up needing a booster before other people do, just to make sure that you're really locking in those long-term protective immune responses.
Thank you. Perfect segue to booster. Carlos, what are your thoughts? Should we all receive a booster? One, because of we have to reactivate our immunity over time, or second, because of potential variants that evade our immunity?
So at this point in time, there is no recommendation of boosters for any group. However, I want to call your attention to a paper that came out just this week in Annals of Internal Medicine, looking at solid organ transplant recipients. And what they did is they saw how much of an immune response they had. And some of those individuals had a very low immune response to no immune response. And giving those individuals a third dose, a booster, actually really boosted their immune response and got a much better immune response, almost back to normal. So I suspect the first group we're going to see some sort of booster on is people who are solid organ transplant or severely immunosuppressed individuals, because we clearly know that they're not as protected, but that third dose appears to have, at least in this paper, appears to have some benefit. I think we're also going to see sort of maybe a recommendation for boosting, and some people maybe have gotten just the Johnson & Johnson vaccine as a way to say, you know, we can increase your immune response. And again, there's a study from Spain using AstraZeneca followed by Pfizer, showing a very good robust response after you did that. So we may see an mRNA vaccine after a vector vaccine. At this point in time, I don't see any other reasons to have a booster in the short term. Now, it may be that in the long term, there's new variants that evade the immune system, or we begin to see, you know, waning of antibodies, in which case a booster will be required. But I remind people that, you know, participants that we followed, granted, they were healthy volunteers, they were doing fine, but people that were enrolled in the phase 1 study of Moderna that I'm very familiar with, who were enrolled in March of last year, as of last week, they still have a very high level of protective immunity. So they're a year out, or more than a year out, and they still have good protective immunity. So I don't think it's going to be, when people are starting to worry, oh, I'm going to need a booster by December. I really don't think so. And I think there's no data to support saying that.
Okay, my last question to you before I open up is, previously infected individuals, how well are they protected? And we get this question a lot about masks too. You know, okay, you know, some of them did get vaccination, others did not. You know, so are they actually in the same category as those who's received vaccination? And what kind of immunity can you expect from them? Angie?
So we still don't have a good correlative protection in terms of protective immunity, meaning we can't just look at your antibody levels and say you have enough that you have this level of protection. So it's really difficult to say. But what we do know is that people who have recovered from COVID have a pretty big range of those immune measurements. So they have a pretty broad spectrum of different neutralizing antibody titers, of T-cell responses, and so forth. Some of them are comparable to people who've had vaccines. Some of them, however, are not. So we do know also that vaccines consistently induce these high titers of neutralizing antibodies. They consistently induce these T-cell responses that are thought to be antiviral and protective. So I think that, you know, to err on the side of caution, I think it's very prudent to recommend that people who have recovered from COVID do get vaccinated. We also have data that shows that one dose of mRNA vaccines is sufficient to really boost up neutralizing antibody titers in convalescent patients to the level of people who've had both shots of their vaccines. So it may be that going forward, people who have already been infected are only recommended to get one shot, because that one shot effectively acts like a booster shot from their initial infection. But the short answer is we don't know. So we're just going to err on the side of caution and say that everybody should have the same level of antibodies and immunity that a vaccine can induce.
But I think what's really important is, again, when you think about many countries, let's say India or other countries that have significant epidemics happening but are trying to vaccinate, I think you can safely say, let's delay vaccination on those that have clear evidence of disease and focus on those that have not. Because again, you know, in some countries, that's 20% of the population, 30% of the population that you don't need to right away vaccinate.
Yeah, I do agree with that. I think that the rate of reinfection is generally low, and there's no data to indicate that in people who are reinfected without vaccination, that they always have more severe disease. In fact, that seems to be the minority, the exception rather than the rule.
Thank you so much. Now let's, I'm sure there's lots of questions. So I'm going to open up to the participants. Casey, are there, first of all, by the way, you can use the raise hand function, which is under reactions. You can also wave to me if you turn your camera on, or you can actually use the chat function to ask questions. Casey?
Yes, one of the questions we received are, what are your thoughts on whether any of these variants might be harmful to children?
Carlos, you want to take it first?
You know, I think again, it's going to be the same whether you're a child or an adult, if your immune system, it's going to be, if it's going to be more transmissible, it's going to be more transmissible. I think the question that is a little more complicated and continues to be an area of significant debate is how critical are children in the transmission and the dynamics of this infection. We clearly know children are infected, but contrary to the flu, the evidence is simply not there, is not clear as it is in the flu, that children are a major reason why the community spread of this disease happens. So I think that the issue in children is, in general, they tend to not get as infected, or at least they don't tend to get as sick as adults. I mean, in this disease, one thing we've learned is the older you are, the more likely you are to have a significant disease and a severe outcome as a result of infection.
Angie?
Yeah, I agree with that. I think that children are already woefully undersampled just because of that. Like, if you don't get sick, if you aren't symptomatic, you're not going to take your kid in for testing. So we really don't fully understand the prevalence in children. We do know that children are susceptible, so variants should be able to infect them just as well as they can infect adults. But I agree with Carlos, we don't really know the significance of them in terms of onward transmission, potentially to vulnerable older people in their households who haven't been vaccinated or who haven't mounted protective responses to the vaccine. So, I think that it is important to continue thinking about ways to mitigate children's exposure risk. And also, while we're thinking about global vaccination strategies, those really do need to include children at home and abroad.
Other questions?
Another question we received is, will we see new variants every year, and will they be more harmful?
Angie?
We'll see new variants every time the virus gets into an unvaccinated or susceptible population. Whether or not those variants will be harmful is really just a matter of probability and luck for the virus, bad luck for us. The way that we can minimize that risk is by minimizing transmission, both through vaccination and in places where vaccines are not widespread yet, through non-pharmaceutical interventions meant to reduce transmission. The virus can't mutate. The virus can't replicate. It can't mutate. If it can't mutate, new variants can't emerge. So the key here is to minimize the number of SARS coronavirus 2 cases overall, take away the virus's ability to replicate, take away the ability for new variants to emerge.
Can you not argue that, given the fact more and more people are vaccinated, that variants are likely to escape immunity, otherwise they won't survive? They can be transmitted.
So yes, you can argue that. However, I'm not convinced that that is a huge risk among fully vaccinated people. And the reason for that is just that we know that these vaccines, even though they weren't tested for their ability to prevent infection, they are actually very, very good at preventing infection altogether. So if you don't have infection, you're not going to get replication, and you're not going to get new viruses emerging out of that really intense selection pressure. And it is really intense selection pressure. The immune system is not just neutralizing antibodies. A virus would not be able to completely circumvent immune protection with a couple of mutations. It would need a lot of changes. What I'm more worried about is replication in partially immune populations. So people who are partially vaccinated, people who've been infected before and have those low responses that I was talking about earlier. That is a situation that would be conducive to both infection and replication and onward transmission. That would not be such crushing selection pressure that the virus wouldn't be taken out by it essentially before it could start to adapt to it.
Carlos, your thoughts?
No, absolutely. I think it's that transmission and replication in partially protected individuals, and also, again, thinking about the global pandemic, is ongoing, replication and transmission in places that it simply is not stopping, right? And all you need is a lot of replication for this virus to have a lot of mutations. It does this naturally. It doesn't need the pressure. Simply the replication, RNA viruses like this one produce enough errors during the replication and the transcription process that you will see the more transmission you occur, the more mutations are going to emerge, and eventually you will see just by lottery, as was mentioned initially, the more robust strain is going to be a Darwinian process in which the more robust will survive and will essentially outgrow the others.
Yeah, and I'll just make one additional point too, that sometimes the virus doesn't need to even evolve with immune selection pressure to become immune evasive. So the epitopes that are responsible, or the parts of the mutations in the spike protein that are responsible for it escaping neutralizing antibody responses, are in the receptor binding domain. Those mutations probably were selected for because they bind ACE2 more efficiently and allow the virus to infect cells and replicate more efficiently, which is really what a virus's evolutionary mandate is to do. It's incidental that those same parts of the spike protein are also important for antibody neutralization. So we don't necessarily even need immune selection pressure to get at variants that are at least partially immune evasive.
Other questions? Yeah, I've never seen such a shy audience. I know many of you.
Victor, we actually have Victor, we do have something in the chat box. So
Okay.
This says, I had an antigen test and was told I had antibodies to the vaccine. I was vaccinated 4 months earlier with Pfizer, and that I did not have antibodies to have been affected. What is the basis of the outcome and how reliable is the result?
Well, let's put it this way. The problem is, so the antibody tests that exist, it's a little bit of a wild, wild west. So many tests were approved, and I think the FDA needs to do a little cleanup on what happens. The great majority of antibody tests are testing for nuclear protein, and the tests that test for nuclear protein will tell you whether you've been infected in the past or not. There's some of the tests, very few, that test against spike protein. Most of them actually test against spike and nuclear protein. If you get one of those tests, you will know that you have antibodies against spike, which will be induced by the vaccine. So a mistake that I frequently see is, well, I want to see if I have antibodies to the vaccine or not. They'll get tested with antibodies and they get the wrong test. They get the test for nuclear protein and they say, oh, I don't have antibodies. Well, you don't have antibodies to the nuclear protein because you've never been infected. You need to ask specifically for antibodies against spike. I wish the FDA will provide some guidance and some recommendations around it, because everybody's getting tested that I know, especially immunocompromised patients. Unfortunately, both the FDA and CDC recently came out with recommendations that we don't test people and that we don't use antibodies to know whether somebody's been immune or not. And that, I think, is creating a lot of sort of people are just doing it outside the FDA recommendation. And I think that's creating even more confusion. But what this person put in the chat is exactly what happens. If you're tested both with nuclear protein and with spike, you'll be able to know whether it's the vaccine or previous infection.
So Carlos, what would you advise James to do? He has to navigate a complex system to figure out what to do next, where to get it, what tests to get next.
Well, I think you really need to see at the place that you're getting, be sure that you're getting a test that has antibodies against spike protein. And there are some certain antibody tests. And for example, some places are beginning to say, like my healthcare system is beginning to say, if you want to check for past infection, you need to order this test. If, for example, we're doing that, and let's say somebody tests positive in the hospital, but they're asymptomatic. Sometimes it's just because they had a past infection. We'll check antibodies on them just to be sure that's the case. And there's certain tests that are for spike protein, but they're not commercially available as such because all of them are under a EUA. And that is, again, what creates a lot of confusion.
Yeah, and adding to that confusion is the fact that some of the manufacturers, the larger manufacturers like Roche, actually make both tests for spike, anti-spike, and anti-nuclear protein. And you have to ask for the right one. And not every single clinician who's going to order these tests necessarily knows to discriminate between those two things. The way you can determine this is by looking at the package inserts, which are available on the FDA's website, but it can be very confusing, I think, for people to navigate. In James's case, I mean, it sounds like the good news is he does have antibodies to the spike protein. So in that sense, I don't think he needs to get another test. That shows that his vaccination worked. And it shows that he probably didn't have COVID before. But this is something that people and clinicians should keep in mind when ordering antibody tests or when requesting them. You can discriminate between antibodies to the vaccine and antibodies to natural infection, but you have to make sure you're getting the right test.
But again, what
James?
Angie also what Angie mentioned before, I think it's really important. Even if you get the right test, you will know you have antibodies against spike, but you don't know if you have enough antibodies to be protected. And that's part of the challenge, right?
We have another
All
question
right.
that came in.
I
Do
think
we
no,
have,
Sorry, Katara, I want to ask James because he had his hand up first.
okay,
James Stan? Now he's got.
Sorry, I had to mute it. So I came in late, and I'm sorry if you've answered this already, but you know, I keep reading alpha, beta, then all of a sudden delta, now possibly gamma. How do we get these variants named? Why?
Well, that's a great question because there's a bunch of different naming systems. The one that was in the news before is called the Pango lineage system. That's B117, B1351, that's beta, P1, that's gamma, B1617.2 is delta. And so obviously this alphabet soup of names was pretty confusing for people. So people often, and by people, I mean also members of the media, would often default to saying the South African variant or the Brazilian variant or the Indian variant. And the problem with that is that it's very stigmatizing for people from those places to be associated with a new variant that's killing a lot of people. And it really can have some pretty bad consequences. We've had a problem with this for years in virology because it used to be the custom that we also named viruses after where they emerged. So the WHO decided to get together and come up with a new system that would be easier for people to talk about, but would, also not be stigmatizing. And the system they came up with was using the Greek alphabet. Now you can debate whether or not that's helpful or not. It's certainly easier for a lot of people than, distinguishing B117 from B1351 or whatever. But I hope, hopefully the pandemic won't get to the point where we run out of Greek letters, because I'm worried if we have to start combining them, the new variants are going to sound like fraternities.
James, last I looked at it, we're up to Kappa.
Yeah, we're up to Kappa.
Yeah.
Oh,
and,
right.
And that's what is better than B117, B1351, P11617. I can't remember those numbers. All right, Takara?
yes, we have another question. Do we have a good estimate of the fraction of the US population that has recovered from undiagnosed COVID infections?
There are some prevalence studies and there is some data out there. And I think you cannot really say of the US population, you may be, if you're sampling in New York City, even in New York City, you may be in certain neighborhoods in the Bronx that it may be close to 50% of people were infected there. But you may be in some places in the Upper West Side that it may be, 10%, 12% or something like that. So it really is very localized. But when you look in general, for example, at a state level, one of my colleagues here at Georgia Tech has developed a website that's called Community, and it looks at the percentage of population calculated to have recovered from infection in that state and combines it with the percent of the population that have been immunized in that state and adds those two numbers to give you the level of population immunity in that area. And if you do those two things, in the US, we're probably around somewhere between 35 to 45% of the population has been infected. And then you add to that, the people that have been vaccinated. Now, many of the vaccinated were also infected, so they're not independent populations. If you add those two together, you get to about 62, 63% at the US level. But if you get to some states like Vermont, you may be more in the 80% range. If you get to some states like Alabama, you may be more in the 25 to 30% range. So it really is very variable. We need more data really, and again, something that really has not been done effectively during this pandemic by CDC or local health departments.
Okay, let's take one more question because I want to ask you both the last question. Takaro?
Okay, the last question is, if I am fully vaccinated with the mRNA vaccine, Should I worry about traveling to somewhere with Delta or other variants?
I'd say no, just because Delta is here. So you don't have to travel to go someplace with Delta or other variants. Those variants are in your community. The question is not, are they there? The question is, how prevalent are they? But in general, if you're fully vaccinated with an mRNA vaccine, you're 2 weeks past your second shot, those provide excellent protection against all of the variants. So while you should, continue to take basic precautions like washing your hands and being respectful of others, that you don't need to be excessively worried if you've been vaccinated. That's the reason why you should get vaccines.
Okay, well, I was going to ask you this tough question about the origin of the virus. No, I'm kidding. I don't want to open that box. But my last question is, how would it end? People talk about this possibly going endemic over time. So, Carlos, you want to go first?
Yeah, it's really complicated to figure out how this is going to end. I think, we're going to see sort of an end in the US in which is going to become, hopefully, as we vaccinate people, we're going to have a little bit of an endemic disease, very much similar to having, the flu or something like that. I think that's where we're going to be. I think globally, it's a different situation, and we may see waves continue throughout many times. I mean, we're beginning to see cases go down in India, but we're starting to see cases go up in Africa. Latin America is having a huge explosion in some countries right now. It's coming down in other countries. So I think it's almost going to be like a balloon that you squeeze it in one place and it blows in that one, but then it goes down in the other one. And until we get vaccines globally, I think we're going to have a continuous, problem globally. In the US, I think it's going to become more of an endemic situation in which we're going to have little outbreaks here and there, but hopefully we will not see again things like what we saw last winter, which was really terrifying.
So, Angie, will we have to get vaccinated every year?
I think that sometimes may be wishful thinking on the part of people who are CEOs of pharmaceutical companies. Right now, anyways, looking at the data, it doesn't seem like we're going to need a booster shot every year. And I really wish actually that the focus would maybe be a little bit less on this and more on getting vaccines out to countries that I think you and Carlos both pointed out earlier have very, very low vaccination rates. I think that is going to be really critical because a pandemic by definition is a global public health crisis. It's not restricted to one country. And I think that even if it, I think it will become endemic in places like the US where the vaccination rate will eventually be very high, certainly is already very high in some country or communities and regions. But I think globally, we need to really make sure that this is a global immunization effort that will prevent us from needing to get more vaccines or more boosters, because that will also prevent the emergence of new variants. And I think that, there's certainly a possibility that we may need to get a booster periodically, depending on when immunity starts to wane. Not every single vaccine provides lifelong immune protection. But I don't think it's going to be annually. This is not influenza. There's only still, even though there's multiple variants, there's really only one species of SARS coronavirus 2 circulating, which is very different from the situation with influenza. Influenza is also transmitted back and forth between the northern hemisphere, southern hemisphere by migratory birds. That's not the situation with SARS coronavirus 2 either. So we really just need to worry about this in the human population for now and focus, I think, on getting the world vaccinated before we start worrying about when we're going to need boosters and how often.
Well, thank you, both of you. Just wonderful, wonderful discussion. Can you all join me and give them a round of applause? Thank you. Alison, back to you.
Yes, just to wrap up, wonderful, wonderful session. We all learned so much. It was really engaging, and we really value your time and expertise and all the effort you're doing to help the nation and the world overcome this this pandemic. And thank you to everyone for joining us. We'll record this and be able to share it going out. because I think it's really wonderful, and we will keep you posted. Look out for our next Tele-Townhall, where we invite you back to learn more about the important impacts of the national academies, and thank you for your support.
Thank you.